the practical starts with a presentation, short presentation, maybe just like 30 minutes, followed by a clarification session. That is the interaction session. You can ask your questions to the respective TAs, and it will be like that. And you will have a quiz. You will have a quiz for the experiment first and two combinedly. That will be just on the next week. Next week after. I mean, the quiz it will be after the two experiments, but you have to same way you have to write a report, and the report you have to just we will give the questionnaire on the next week. So once after finishing the last two experiment, I mean the first two experiment, you can write the report. So and the other thing it is your attendance also will carry marks. So make sure you attend all the practicals from the entire session. If you are leaving in between, and I think so you may lose the attendance mark. So, so any clarification? If you want some questions, so you can ask me. One more important thing, and your theory class. So this time, Professor Annab is going to take your theory class. It will start from the next. I think it is Tuesday. So we'll have theory class on Tuesday and Thursday as usual for just like the other modules, immunology and molecular biology. And timing is 12 to 11 to 12, I guess. Yeah, I guess 11 to 12. Okay. So soon this PhDs and I think Arnab Nandi is going to give you a presentation about the neuroanatomy. So just let's wait for him.
Yes, Aditya, you have queries?
Yes, sir. Sir, you mentioned that attendance will carry marks. You said that if we log out in the middle, then we might lose our attendance.
Yeah, sure.
It so happens that my connectivity, as well as a few other students' connectivity, is not as stable as ideal. So it happens that at times I need to switch from one network connectivity to another. So for 2 to 3 minutes of session, I tend to log out and then come back.
That is fine. In the connectivity loss in between your practical module, that is okay, fine. So anyhow you will come back, right? So if you are leaving and just for the entire session, then you will lose up attendance.
Okay, so amount of time attended for the session that determines the attendance, right?
Yeah. So for the just like until the end of the session, you should be there.
Yes, and will there be any a roll call for our previous?
So that I'll just you can ask this question to Vinod. Vinod, so how you're taking the attendance? Any roll call or something for the other module?
No, I can take a digital form of attendance. Whatever time lap, if they are left even for 1 minute also, I will get a notification each and everything. So based on that, we will give them attendance.
Yeah. Not everything. So who are living in between or something. So the details. Yes. So we'll give the attendance.
Yes.
Okay. So I think still the Arnab has a technical issues or something, and
no, it's okay. Now it's connected.
Now it is connected. Okay, Arnab, you're only giving the presentation, right?
Yes.
So who is there in your team for this clarification and interaction? Garima and who else?
Aishwarya.
Aishwarya is there. So yeah, I think I think three more to join or two more to join. Vinod, can we start now?
We can wait.
Yes. We can wait 5 minutes.
We can.
Huh?
Yes, we can.
Or we can wait for 5 minutes.
We can wait for 5 minutes.
Okay, that is fine. That's fine. We can wait for 5 minutes. I mean, 2 or 3 are there.
Maybe network issues like ours there.
Yeah. I'll let them join.
5 minutes is fine.
Yeah, 5 minutes fine. So for the remaining, if you have any doubt regarding the structure of this module, so you can ask.
Sir, we'll be having a quiz from the next week, right? So
you'll have just like a completely once after finishing your your two experiment, the first two experiment, you will have a quiz. Okay, so that time is not fixed. So based on your convenience, for everyone convenience, just we will fix that one. But for the next two, I mean, the last two practical, that is the memory and cognition, that regularly will have a quiz. At the end of the practical, they will send you questions. Will have quiz. For these first two experiments, the quiz will be after the just like all the two experiments we finish. Okay.
Thank you, sir.
Sir, about the lab reports.
Yeah.
So,
Yeah.
yes, sir. So let's say the lab 1 is done this week.
Yeah.
So by next Monday, we have to submit the lab report, is it?
No, it's not like that. Okay, so just it's not like that. Okay, we'll give a questionnaire on Monday. That's why we are planning like that. We'll give a questionnaire for your lab reports. So that will give on I mean maybe Friday or Saturday. So you can finish your lab reports once both labs are finished. First and two labs. Once it is finished, you can submit the report. We'll give the deadlines. Okay.
Okay. Sir,
So will the format of the lab report be specified when it is given to us?
Yeah. Just we will give the, I mean, what are the things you have to do? Like maybe just like questionnaire, what you have to write. Okay, we'll just send you that one. Okay.
All right.
Sir, actually, I might have joined few minutes late, but we are supposed to submit all the lab reports together at the end of the module, right? Or like
No, it is
individually?
so actually we have four experiments here. Then the first two part is just like a I said neurobiology or something. That two experiments is different. So they are just like so that we will do separately, and the other two experiments, the last two experiments, this just based on the using MATLAB and this neuron software. So that is I mean it's a different module. So that assignment and it will be different. Okay. So we are planning to give it in a separate. Okay. So there, for the last two experiments, there will have quiz the same day of the practical and the report also like that. Okay. So we'll let you know.
So another thing, like what will be the weightage of like, and is there going to be any viva or like at the or an exam at the?
Yeah, you'll have this exam. That is, I mean, you'll have an exam for the first two experiments combinedly. So, and there is no, I think so far we are not planned any viva, and I think it's so we'll avoid that one. I think we don't have any viva.
And so another thing, what will be the weightages of each of these quizzes or weekly exams and the lab reports?
So weightage, just we'll give the details of the weightage. Okay, means so how the attendance is the must, and so we can give weightage, maximum weightage to your quiz, then followed by this assignments, then attendance.
Okay, thank you, sir.
So you said the first two labs will have like one lab report. So is it a condensed lab report of both the labs, or is it like two different lab reports?
It's two different lab reports.
All right.
So, the exam will be same for these two. So the skills will be combined for the two experiments, but report will be different.
So should I start, Jim?
Yeah, you can. I think
Yes, you can
yes,
start.
you can start.
Because I think they are not joining.
Okay, so Annab, you can start. Okay, so this is Annab from neurobiology, and he's going to give the presentation, and along with Garima is there and Aishwarya is there. And after this presentation, you can I mean, you can ask question to Annab and this team. Okay, so they have been doing this one, and they are very. I mean, they have specialization in this neuroanatomy and all, so you can clarify your doubts. Okay, Annab, you can start.
Okay. Yeah, everyone can see this? Okay.
Yes, okay.
Yeah. Okay, so welcome to the neuroanatomy class. So uh, to start with, what we'll give you is a summary of the nervous system and why it is important to study the nervous system itself. And we also walk through several points of main points of the this practical here. So in general, you can define like a nervous system as like there is a system that is there. It gets an output and it gets an input and give a output like a function or like a machine. So in biology, for input you have sense organs. Uh, you have your skin, you have ears, you have nose, and the sensory nerve. Yeah, you have sensory nerves and tracts. This conveys to the system, which is our central nervous system. And after the central nervous system, via the motor tracts and nerves, it goes to the motor effectors, and then you act. So it could be seeing and snake, and then jumping to move away from the snake. So there are a lot of things. So neuroscience is a very interdisciplinary subject. There could be physicists, mathematicians, from computer scientists. They can contribute to the neuroscience field. There are also psychologists, which you might expect that they will contribute, and also philosophers and biologists as well. So it's a very interdisciplinary field, and you can study the brain in different from different aspect as well. So early development of the nervous system, if you go back, the single cell organism didn't have any nervous system. When they become multicellular, like the sponges and organism, there becomes a few specialized cells which gets their inputs and give the outputs. After which there is a format of the nerve net first in the hydra, and their neurons form a nerve net structure, and there is input and one interneuron in between, and there is effector cells as well. So during development, one of the most important thing is in bilaterally symmetrical animals is the cephalization. So there is aggregation of this all this neuron, all these sensory cells in one part of the body. You can call it, for example, here is the brain, not really brain, but a cephalization in a way, or the centralization. In the leech as well, you can see one cephalization here, even in the arthropod and the squid. So if you observe that they become more cephalized along with the phylogenetic tree. So these are different brains of animals. So can you say that which are the species they might belong to? This one? Or which one is human, you might think?
Sir, I feel the bottom one is human. The bottom one.
Okay, and this one maybe?
It might be a different angle of the human brain, maybe. That's it.
Okay. And this one?
Is that a bird's brain or something?
Okay. Okay, let's see. So this one is a rat.
I'm guessing the second last is the okay. The last one is a human. Okay, fine. My bad.
Okay. So this one is a rat brain. This one is a cat brain. This one is a dolphin brain. This one human. This is chimpanzee, and this is sheep, and this is rabbit. Look that you know already that dolphin is very intelligent animal, right? So notice that their size and different proportion of the regions here. So we'll talk about this as well in this presentation. So there is a increase in this brain size. So this part is optic part. It's very prominent. Olfactory part, olfactory lobe. It's very prominent in the rats, but it becomes smaller because rats are very dependent on the sensory inputs coming from the smell. So their large part is dedicated to the smell here, compared to the other animals which do not rely much on the sensory inputs from this. Whereas as a visual system, if they are like the chimpanzee or this chimpanzee or the sheep, they are more relied on this visual information to look for their enemies here. So this part is bigger compared to here, the visual part. And there is additional with human regions which are more specialized and become got bigger. So there is an increase in the brain size along the phylogenetic here. So before we go to the anatomy of the brain, we first need to know that what axis we have for the anatomy in humans and in other animals as well. So human, you have to know it differently compared to the cat here, which is four-legged, and human two-legged or bipedal. So the axis you will see will be different here. So this is a posterior anterior axis from front to back or back to front, and perpendicular to the anterior posterior there will be a transverse plane here. This is medial or lateral, and the mid sagittal which divides from the nose to toe in half, and the head is the superior and the bottom is the inferior. Like the cat here, the nomenclature changes a little. So the dorsal at the back and the ventral is the bottom part, and the rostral is the front, and the caudal is the tail. Here the example of the frog as well. So from the horizontal or the sagittal. So compare that with the brain axis now. So this brain axis, so the anterior part is this so you are viewing it from the top. So say this human, you are viewing from the top of its head, and so this is a dorsal view. You are seeing it as if it facing towards the north or the up. So this is the anterior posterior axis. So the center one is the medial or this medial. So this is the near the midline here, and the two away from these two are the lateral. Now compare this with the rodent or this mice or rat here. So there is anterior is there where the nose are, and the posterior is the caudal where this tail is situated, and this dorsal is their back, ventral is their bottom, and it's their brain here. When you view from the top, the midline is which actually divides it in symmetrical half, and the spinal cord goes along the midline or the medial here. Now, so there is a concept of ipsi and contralateral. So ipsi means on the same side. So say if I say one region is ipsilateral to the other, so say this front region is ipsilateral to this back region, means situated on the same side, while the contralateral is one region just opposite a symmetrical region or in the other side. So this is called a contralateral region. Okay. So now going to the anatomical coordinates of the brain. So if you cut the brains in different orientation of this axis, so you will get different types of section. So if you cut if you cut from top to bottom or the dorsal to ventral perpendicular to the midline, you will get a section called coronal section. And this is named after the corona of the sun during the solar eclipse. If you cut from anterior to posterior perpendicular to the midline and top to bottom, you will get a section which is axial. And if you just divide it in half from the midline, you will get a mid sagittal section, which looks like this. So these are just different planes in which we'll be talking about the human brain as well as mice brain as well. So the nervous system is broadly divided into peripheral nervous system or the central nervous system. We all know from your class 12. And the peripheral is subdivided into autonomic and somatic nervous system. Autonomic, which is further subdivided into sympathetic and parasympathetic nervous system. So for example, the sympathetic nervous system is when you see like a snake and you respond. So that's a sympathetic, when you fearful response. And the central nervous system here is consist of your brain and the spinal cord. And the brain is further subdivided into your front part, which is the forebrain, or the mid part is the midbrain, or the back, which is the hindbrain. These are further subdivided into different subregions. Example, telencephalon or diencephalon, depending on their region, and again subdivided into different specific regions as well. For example, cerebral cortex, basal ganglia, hippocampus. So you can see the cerebral cortex from outside, but this basal ganglia, hippocampus, or amygdala, you have to cut open the brains to see these regions, as well as the thalamus or hypothalamus. So these regions you can see. So that's why the need for the axis and the cutting the brain comes. And you will also see the for the metencephalon part there is pons and cerebellum, and you will also see the medulla part. So this is just broadly divisions of the nervous system. So during development, how it is developed is that you have ectoderm layer, and it forms this epidermis and the neural tube, and it further the brain and other regions as well. And this neural crystal forms this peripheral nervous system and other cells like the melanocyte or the adrenal medulla cells here. So during the development, it starts with a tube and it bulges out and it divided into 7 regions with their time during development. This is the same thing. Yeah. Yeah. So this is a real photograph of human brain. So this part, which is you see here, this is called the cerebral hemisphere. So can anyone say which plane the brain is cut here?
Mid sagittal.
Okay.
Yeah, it's a mid sagittal plane cut. You see this white region here? So this is called corpus callosum. This holds together the two parts of this cerebral hemisphere of the brain. And you see there is a leaf-like structure here. This is called a cerebellum. And the medulla part is like in the brainstem here at the end. You have a region called pons here, a bulging part here, and the midbrain structure is this one. So formally, the cerebral cortex you can call is like a telencephalon or different regions. These are the informal division and these are the formal division part, just the name of the same region. So our brain is situated and protected by our skull, and the skull has different bones. And after the name of this different bone, the region of our brain is named after those. So there is bone which covers the part of the brain which is called frontal lobe. And this part here is called the parietal lobe. And in the back, you see occipital lobe. And here, the green part here is called the temporal lobe. So all of this lobe perform different functions in our brain. And again, this is another example here, is the cerebral hemisphere. So you see here, there are groups and there are some ridges as well. So the groups that are there and the ridges that are there, they're named differently. Anyone know which are called what? The groups and the ridges?
Sir, are they called the meninges?
No. So the meninges is the covering of the brains. What I'm talking about is you see this one, there are depression or this groove that are there, and there is hill or the ridge like region.
Sulcus and Gaia.
Yeah,
Sulci
sulci and
and
gyri or
Kyrie.
sulcus or gyrus. Yeah. So there are different here. So this part here in the just above this temporal lobe here, this is called a lateral fissure. This big groove here. This ridge here is called a precentral sulcus, and this ridge here is called the groove here is called the central sulcus. So this groove, and again this ridge is called post-central gyrus. So central sulcus, pre-central gyrus, the ridge, and post-central gyrus. Again another ridge after the central sulcus. So you have different regions like you saw in here. There is brainstem, there is cerebellum, and spinal cord here. It cut here, and there is also this occipital cortex, and here you have this preoccipital notch. So, hmm? So brain is dedicated differently for the sensory response. This is called a homunculus. So the sensory responses has more input from our different parts of the body. So for example, lips, eyes, and the tip of our fingers, these are more dedicated, have more dedicated regions in the brain than the other. So this is for the somatosensory. So only sensory part which are more sensitive inputs to brain, and this is motor. So which part of the body has more motor input output from our brain. So this is a cartoon depicting the same. So the outer part is here the cerebral cortex, but we have a deep brain structure which you only see when you cut the brain. And this part is called the limbic system, which controls mostly our emotion and the emotion. And so this central part is called the thalamus. And this bent part here is called the fornix. You have this thalamus, below the thalamus, you have something called hypothalamus, which is responsible for our stress response. You also have this region called amygdala, which responds during our fear. You also have a region called hippocampus, which helps in formation of memory and maintaining our memory. So in the different parts of the brain, we have different regions here, like the hypothalamus. And this brainstem part is our respiratory center. It controls our respiration. And you also have this cardiovascular center here in the brainstem as well. So this brainstem part control our respiration and cardiovascular So we also have a spinal cord here, which is a part of the central nervous system, and you have this different nerves coming from the spinal cord here and going in, both sensory and your motor, and there are interneurons which regulates our reflex here. So compare that with the section of brain and the spinal cord. You will see here this region in the brain is lighter color. This is called a white matter, and the dark color outside is called a gray matter. If you see in this spinal cord, this become reversed, and this is because they cross over in our neck. So you have different nerves here and different in the spinal cord coming from this different cervical or the thoracic region or this lumbar region or the sacral region named after this different bones in the spine. So our brain is protected by the skull, but it also protected by a covering called the meninges. And the outer part of this meninges is the dura mater, which is the toughest part, and there is vascularis part inside, which is called araconid matter, and which will have their blood vessels as well. And the thinnest part is called a pia mater. So this is an electron micrograph of the meninges showing different layers. For example, this dura matter or this araconid matter and your pia matter as well. And here starts the brain tissue. And this is the blood vessels that supply the brains. So in the brains, to supply for this, you have ventricular system, which contains your cerebrospinal fluid, and it supplies nutritions and take away materials back into the blood. So this part here is called third ventricle. This part is fourth ventricle, and these are different regions of the same ventricular system.
Sir, I had a doubt. What is
Yeah.
the purpose of the ventricle system?
This is so brains to supply the nutrients in the brains.
Okay.
Generally. Yeah. So you also have this arterial blood supply here from outside as well. And this goes in these groups of this sulci. This is a comparison again for the rat and human brains of different section wise. So this is the mid-sagittal section of the rat, and this is the mid-sagittal section of the human brain. So now compare the size of the cerebral different region. So the cerebral hemisphere here is, and this one, you see there is no sulci here in the rat brain, whereas humans have a very much this sulci and gyri here. And look for the differences. So this optic lobe that you olfactory lobe that you see here, it's gone in here. You don't see it because you're not dependent on the sensory input from the smell much as the rats do. So these are some suggested readings you can look for. We'll send this PPT to you. For the you identify some regions here? For example, this one.
Corpus callosum, right?
Yes. And this, the whole region.
Parietal.
Yeah, you are correct. But actually, I am referring to all this. But yeah, this is a parietal. And this part.
Cerebellum.
Yeah. So this is the end of the slide. Yeah. So yeah, have you gone through the video there? Easy guys.
What video, sir?
Anna, did you upload any videos for this one, for the brain sections and the sections we have? No, just like we took videos, no? That is not that uploaded, I guess.
Oh, that video is not still uploaded.
Yeah. So you
Oh,
said
okay.
like that you will upload today and you will give an explanation or something?
No, no. So I sent to Binod already, I think.
Oh, Vinod. Hello.
Yes.
Vinod, that video he has sent to you for the brain sections they prepared and the
Yeah.
short videos. Can you upload that one?
Yeah.
So,
Sir?
Yeah. Anyway, who
I
is
had
having question? Yeah.
I had a question. You mentioned about the articular space, right? And then next to that, there was some subarticular space. So, what is the composition of subarticular space?
It is not subarticular, subarachnoid space. I think you are referring to.
I
Wait. I'll show you.
Link is in the chats. Arnab.
Yeah. You see this one you are
yeah
talking about.
yeah yeah. Sorry, my bad.
So this one has your CSF or the cerebrospinal fluid.
Oh, okay. Okay.
so Vinod, you have this, upload the link.
Yeah, you all can go through the video once.
So Anna, 1 minute, just I think they haven't seen this video. Can you play that video on your system and share that screen so that everyone can view right
Yeah,
now?
yeah. Wait.
Hmm?
Yeah.
Arnab, you can share the screen with your computer audio. Then
Yeah.
audio is.
Or sir, can we view the video on our own and then come back after 10 minutes? Because there's a lot of lag which we are seeing.
Yes, sir. There'll be a lot of lag happening.
Yeah, yeah.
Doing it on the same computer.
It's just a 2 minutes
So
video.
you should.
Oh, it's just only 2 minutes video. Just
No,
then
sir.
again log out and is coming. Then again it will take to just come back. No, what do you feel? You want a break just for 5 minutes?
Yes,
No,
sir.
we'll upload it anyway, so don't worry.
Yeah, anyhow we'll upload. So if you want, just Arnab, we can give them a 5 minutes break
Yeah.
from this one.
Yeah, we
Feel
can
like-
be on the call on mute and we can view that and come back in 5 minutes.
Yeah, yes, that's fine, I guess. Okay.
Sure, sir. Thank you.
Okay.
So Binod, are you uploading the video now?
Yeah, I uploaded already. It's in the
Oh,
chats.
okay. You are sharing. Okay.
Yeah.
Thanks.
So can you explain how to install Neuron on MacBook? Hello.
Yeah.
Yes. Can you explain how to install Neuron on MacBook? It seems that there is a problem.
So although it's not really our part, but I think you have to install VMware to install the Neuron. Like you have to virtually install Windows for that.
Okay.
You know about VMware or?
Yes. Yes.
Okay, then. You can install like virtually the Windows and then run the Neuron probably.
Is there any other way we can do our experiments without doing that?
I don't know because that part is really not what we are going to take. We're only taking the neuroanatomy part here. So that part will be taken by cognitive people from cognitive neuroscience. So they
Okay.
will tell you exactly how to install and everything.
I did not find a problem installing Neuron Vikalpa.
Oh, for me, it just doesn't open, saying that it's from a third-party website or something like that.
Yeah, so you just need to change your settings to
Yep.
it did that to me too. So I just had to unlock my settings thing into
Yeah,
like
it's
make
there in that
Cool, cool.
system preferences, no?
I'll get in
yeah
touch with you after
yeah
the
yeah
lab.
sure, sure. Yeah. Also, sir, let's say if not for COVID, would we would we be handling the brain and then like touching all the stuff in the lab?
Yeah, if it was not, it was supposed to be real practical. So
Yeah.
Oh,
So I
like
think,
the one you are seeing is the same video. You can touch them and see them
yeah, even
and check the video.
another section is there. No, just you are storing that in formal, and the section is there, and yeah, I think it is there, right? No, you are storing that one.
Yeah, everything is
So,
there.
so once after this situation is just gone, if you are interested, you can go there and you can
Yeah,
see and
you can
just
come.
visit. Yeah.
okay, sir.
So it's better to always have a closer look, rather than seeing in videos. But unfortunately,
Yeah, Sam. So he asked how to know which section is to take for which purpose. So for this, if you need a deeper brain sections or a region which you cannot access or you cannot see in same way, so we have to take different way. Like one, if you take mid sagittal, you will see corpus callosum in a way, and if you take a cardinal section, you will see in another view of that.
And sometimes if you have to track the neuronal uh, axons and other fibers, so that depends like how from where to where the fibers are going. So for that you have to take different sections. For example, some will be clearly visible in the sagittal section if the structures are connected like that. So it highly depends on the connection you want to see or the structure you want to see. So that decides the plane of the section.
Also, could you explain like which all types of sections are there? Like
So
you
coronal,
have
sagittal,
mentioned.
and axial sections were there in the video. Same way for the mouse sections also, we can do coronal, sagittal, and other planes. So it depends on the question you are asking.
Okay.
in the like in the video you had mentioned there's a sagittal plane, so that's basically parallel to the mid sagittal, right?
Yes.
What
So
is
And
mid
message?
sagittal is just a mid line, and parallel to all of that, mid sagittal is all sagittal.
yeah, and you had mentioned something in the axial section. I didn't get that.
So axial from when you cut perpendicular to the let's say from head to toe, you have one string, and you're cutting from the perpendicular to that. So I want to show the. Wait a second.
Actually, you mentioned some ventricle in that region. Is it some major ventricle?
Hmm? Oh yeah, yeah. So that's the yeah, yeah. So this one is yeah. So that one will be your third ventricle here. This one. When you cut it, you see this part. So you go to one website. I will tell you. So in this one, you go to this brainfacts.org, and you will see one 3D brain simulation. So you will see one.
So you can
So
just
in
play
there you
with
can
the
yeah.
brain and you can check each and every region. It's in the 3D uh, depiction there.
Okay, and what is the coronal section?
Yeah, so the coronal section is from, say, from nose to and the back, your face to back. You're cutting from top to bottom. So I was going.
So
It's
it's
better to
kind
show.
of dorsal to ventral axis of the animal.
Okay.
Let me show you again.
Sir,
Yeah.
most of the words I'm listening today are I have whatsoever like not listened to them ever. I have no whatsoever foundation
Oh,
about
okay.
this. So could you just please share the slides?
Yeah, we'll share one.
Yes, sir.
We'll share.
We'll share the handout after this thing.
Yeah,
So
this
tonight we'll
presentation.
share one handout.
Yeah.
Yeah, don't worry, everything will be there in the
And yeah,
handouts.
whatever the handout, it will be there. Everything.
Okay. Okay, fine.
So the kernel plane you see here.
It's from.
You understood what is a kernel plane?
Yes, sir. It's basically the dorsal plane
To
itself,
ventral.
right?
Yeah.
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 But dorsal to ventral, perpendicular to your anterior posterior axis,
Ha, yeah.
not the mid sagittal plane.
Yeah, yeah. Okay.
Exit.
Thank you, sir.
Yeah.
ये
Mid
question है
sagittal to perpendicular. What is the question? What is the correlation between?
न्यूरॉन का वो बताना है
Huh.
complexity.
Elephant has big brain, but not as well. Okay. Yeah. So that question. Yeah. So the correlation between this cranial capacity and cognitive ability was saying, although the elephant has a big brain and not smart as human, is not really. It depends on your cranial capacity itself. It also depends on how much connectivity you have, how much neuron capacity you have. So it's all that, and also you compare the size of the elephant as well, and its brain relative size. So mostly it depends on the capacity of the neuron in itself. It's not the how much number you have or the how big it is. So there is a general trend, but not as much strong as like strong. It's not a strong correlation.
Now, is it as something not just related with the foldings, and if you have so many foldings in the your cerebral cortex and all, you seems to be
Yeah,
more.
that you can say. The cerebral foldings also contribute.
So yeah.
So does it mean that the cognitive ability is directly proportional to the surface area of the brain?
You can say, but at the same time, the elephant has like the big brain. It also has a bigger surface area as well.
Oh yes. So
So can you say that? So it's not really just the capacity.
Highly
It's
depends on the connection
yeah.
that are formed.
Sir, could
It's
you repeat
connection, it's synapse, it's proper functioning,
And other cells that are
everything.
helping in
So
the
it's
proper.
still not very much like known or well understood. That's why we're still studying to answer that question.
Okay,
Sir,
sir.
once could you repeat again how the size of the brain determines how smart you are or how not smart you
The size of the brain has nothing to do really.
are?
So say for example, one people may be very smart at like say young age, one small child, they are very smart. Whereas it's possible that you have a bigger brain size at when you become old, your cognitive ability goes down. So your size is still different, but you cannot perform cognitively better.
Yeah. Another one thing, very recently I've seen a interview. So it was an interview based, just like a discussion regarding the Einstein, and in that discussion, one neurobiologist was there, and this interviewer asked like that whether he was studying the Einstein brain, it is preserved,
Mm.
and they were asking like that, whether he was able to find some remarkable things in his brain. He was answering like that he couldn't find anything remarkable. Just apart from that, he just so he found some just prominent area where so people use string instruments. So they have some just like a brain specialized area. Apart from that thing, nothing just like specialized from the others. Like so,
Yeah,
yeah.
overall, like in the gross level, you don't see anything
Yeah, that's the thing. Like seeing
different.
the yeah.
It's more deeper into that instead of simple number and the capacity.
So just like a viewing or something like a size or this things we cannot say like that. You how much brain you can say how much intelligent you are or how much brainy you are.
Yeah.
Yeah.
So what you need is a functionally active and everything, not just a structurally big.
If we have some so many neuronal network connections and all, just we tend to be more intelligent than others, right? You're
Yeah,
saying.
that you can say. That you can say. Like better connections will make.
So you Arnav, you're saying like that we can gain in your lifetime, right? Like this neuronal connections or synapses. So just like then the brain plasticity is something like you can just increase your synapses or networks in your lifetime, right? So
Increase in synapse.
yeah, means these new connections. No, just we can
Yeah, but
it will
so
form.
sometimes you will normally with learning you may get increased synapse, but
Mm.
after when with your age you lose synapse as well. So there is a decrease decline with age.
Yeah, but this new connection can form no, in your no, full lifetime. So sometimes if
Yeah,
you are not
they can
using that,
form. They can form.
Yeah, because of the learning.
yeah, learning. So if you are not using that just neuronal network, so you are not just like
They will
uh,
become weaker.
they will
The
become and they will
synapse will
increase.
become weaker.
Yeah.
So suppose if the connection are formed, they has to be like used. But if they are not in use, slowly the system will kind of degrade those synapses
Yeah, yeah.
if
So
they are not in use.
yes. So
But the
the
synapses which are in use, they will become strengthened over time.
yeah, that's it. So the what is the only way to just like recollect things and do it again or something? Your experience it will make it the synapses so strong, right?
Yeah, I think that's what probably happened. Like it's a speculation. That's probably happened with like a memory that you want
Yeah,
to forget.
yeah,
So you want to forget, that's why you remembered again and then you don't forget. Like it's a mind,
yeah,
just speculation.
yeah.
Okay, so rich connectome means a higher cognitive ability, is it?
In a way. But defining that just a connectome as well. So the connectome is still, it's not like functional connectome when you make it. So it's still structural connection.
Yes.
So the synapse that you see versus the connection that you see normally, so all two things are not exactly the same. So they may be forming the connection, but it's not really useful in terms of, let's say, synapse. So then it's not better.
Okay, okay. Thank you.
So these relations are not very like straightforward that you will have more capacity or more synapse and everything. So these are specific to some the query that you are asking. So the cognitive ability, when you say, it's not all the cognitive ability, you're just testing only one or two aspects of this. So you cannot say that with this we can answer all the questions, right?
Yes, sir.
So yeah. Any other questions that you have, you guys?
Or you can go through the video again.
Yeah,
If you
you
have
can
any
go.
questions, you can
Yeah.
always come back to us and like you can mail or text.
So everyone has gone through this videos and presentation, I guess. So Anab, so they don't have questions, I guess.
Yeah, it seems it is not
So
much of the thing. Also, it's small part.
yeah. So can we wind up the class or what?
Yeah.
So then,
Later. We
so
can if
can
there are no any other like questions.
okay. Hello, students. So just like if you don't have any questions, like right now, just we can wind up the session. So after this one, you just once again go through these handouts and this presentation and video. If you have doubts, just you can mail at any time. So we are here, just like, and the PhDTs are dedicated for this practical. They are there to help us. So write to me or them, so they will clarify your doubts. So yeah, that's all. Another thing, I think tomorrow you have a class for the theory, neurobiology. So please do attend the theory. So just then you will get some more ideas. I think, I don't, yeah, the same syllabus is going parallel to this one. The practical is there. Okay, then so Vinod, hello. Think yeah, is not there. Okay, then Anab, then thanks for Anab and
Yeah.
Aishwarya and Garima. No, yes, I mean I'm not able to see the things. Yeah, please. Okay, then we'll wind up the session right now.
Okay.
I'll stop the recording.
Yeah. Okay. Thanks for everyone for
Yeah.
participating. Yeah.
Thanks everyone.
Yeah.
Sir, won't you in attendance?
Yeah.
Sorry.
So the attendance just we can
Yeah, we
take
took.
down.
I took.
Yeah, we took. Yeah. So we downloaded all these things.